Thanks a lot. So hello, everyone. It's my pleasure to be here again. This is my third year doing the DLRL Summer School, and I really always enjoy it. I think it's a fantastic event. I hope you're enjoying it. I do know you must be tired at this final afternoon of the deep learning session. So of course they gave you me, who's probably outside of your wheelhouse and therefore very hard to understand, and then Joshua, who's always super easy to understand. So pay attention. Okay. So today I'm going to be talking to you about deep learning in the brain. And that might seem a slightly weird thing depending upon your background, because most of us probably associate deep learning exclusively with artificial intelligence. And that is a reasonable thing, because that is where deep learning has really taken off. But it's maybe something you don't know, that depending upon how much you've studied the history of neural networks theory, that in fact, a lot of the research into neural networks and deep neural networks has always been with an eye towards understanding our own brains. And many of the models that were developed, especially in the early days, less so now within the commercial sector, but in the early days of neural networks research, many of the models were developed to understand what happens in our brains. So in this session, what I'm going to do is I'm going to give you a brief overview as to the challenges of linking deep learning with neuroscience, and try to give you a sense of what some of the work in this area has uncovered in the past, let's say, 5 years. Okay, so the first thing that I want to touch on, and I apologize to those of you who maybe have some neuroscience background, because this is going to be super duper introductory, because I want to make sure that people who come from a purely computer science or engineering background are on board. You know, we call neural networks precisely because they are intended to be an abstract model of what happens in our own brains. So our own brains are composed of many different cell types, but the critical cell type for information processing and computation and the control of behavior is the neuron, which you've probably heard about. And on the top here, we have a cartoon version of a neuron, so here, can you see that? No, that doesn't come up there. To the organizers, if there's a laser pointer, that would be awesome. But in the meantime, I'll do without. Sorry, I forgot to bring mine. So at the top here, you see a cartoon version of a neuron. The soma, or cell body, is where the nucleus sits, where the DNA of the cell sits. And one of the things that distinguishes neurons, thank you very much. The thing that distinguishes neurons, there we go. You can all see that? Okay, good. One of the things that distinguishes neurons from other cell types in your body is that they have this remarkable tree-like structure, which you can see illustrated here. And this tree-like structure is key to their ability to integrate signals and compute things. Now, there's two forms of branches, as it were, to neurons. One are called dendrites, and the other are called axons. Now, every neuron has only a single axon, and the axon is the branch that is responsible for sending information out to other neurons. So they have just one output line that will then communicate a signal to all the other neurons that they're connected to. The dendrites are the part of the neuron that receives the signal from other neurons. And basically what happens, I don't have time to go into the depth of the biology because that's a whole introductory class in neuroscience, but essentially what happens is when signals from the axons of other neurons arrive at the synapse, which is, or at the axon terminal, which is where we find a synapse, and the synapse is the connection between two neurons. So you'll have a synapse between the axon of one neuron and the dendrite of another neuron. When a signal is sent down the axon, arrives at the axon terminal, and crosses the synapse, that crossing, by the way, is done by the release of a chemical, usually, a neurotransmitter. When the neurotransmitter is released at the synapse and picked up on the dendrite, it causes a depolarization or a hyperpolarization, meaning a change in the voltage of the neurons, dendrites. And those changes in voltage across a branch can summate. So you can actually show, like experimentally, that in standard conditions, but there's some interesting conditions that we'll talk about where this doesn't hold. In standard conditions, what happens is anytime a signal arrives at the dendrite, all of the signals that arrive on the dendrites will sum in a close to linear fashion. So the voltage impact on what we call the postsynaptic neuron of all of its inputs is roughly equivalent to a sum of those inputs. And that's why when we look at an artificial neuron from an artificial neural network, its first component is just a linear sum of its inputs. That is intended to be an abstract model of what happens in real dendrites. And then the activation function that we use, whether it's ReLU or TANH or whatever, is supposed to mimic the activation of the axon. And again, there's all sorts of complexities that I'm not going to be able to talk about, but I will say that it's not actually that crazy simulation of what happens in real neurons. And you can show that you can actually capture a majority of the variance in what goes on in a real neuron with a simple linear, nonlinear function just like this. So artificial neurons in artificial neural networks are an abstract and not too unreasonable simulation of what happens in real neurons. And then what happens in our brains, of course, is that neurons are assembled into a gigantic network. And in particular, in the mammalian brain, these networks have a hierarchical structure. So here you've got a picture of a macaque brain. And all these regions here, these compose what's known as the ventral visual stream in the macaque brain. This is the part of the brain that is responsible for taking in visual information and converting it into abstract, categorical signals, as it were, that the animal can use to do things like respond to another animal or eat food, etc., identify food, etc. So the information from the retina comes into, at first, a region of the brain called primary visual cortex, or V1. And then the information gets propagated down this ventral visual stream in a series of processing stages that's illustrated here. So here, this is RGC refers to retinal ganglion cells. The retinal ganglion cells project to a region of the brain called the lateral geniculate nucleus, or the thalamus, which is embedded in the center, and then that gets projected to primary visual cortex here. And we see these sort of stages of processing throughout the brain. And what we see is kind of an accumulation of more abstract features. So in early layers of the ventral visual stream, what you find are things like edge detectors. Neurons will respond to oriented edges or moving edges, stuff like that. As you move up the visual stream, you'll start to find in places like V2 and V4 neurons that respond to corners or whole objects, particular shapes. And then when you get up to a region called inferotemporal cortex, so that's what IT refers to here, and these are different parts of inferotemporal cortex, posterior, central, and anterior, you start to see neurons that respond to particular types of objects. So they will fire when the animal is looking at another monkey, or an apple, or a car, literally, whatever. Now, part of what is interesting as well is that the way that these regions are laid out in the brain is they have what's called a retinotopic order, which means that the feature detectors have a particular location in space that they are responding to. So one of the things that you may not know, because this isn't always taught but maybe you do know, is that convolutional neural networks, which is what's illustrated on the bottom here, were originally invented to try to mimic this structure of the ventral visual stream. So when you look at a convolutional neural network, the idea is that in the early layers, you're doing something like what happens in primary visual cortex. You're taking this tiled, did my mic just turn off? Anyone have any idea? Okay, I'm just gonna talk loud, and I'll let these guys figure it out in the meantime. Luckily, I am really loud, so much to my wife's chagrin. Okay, so the structure of convolutional neural networks, like I said, was in fact originally designed to mimic this process. And what we find, sorry, there we go. Hi. Part of the idea as well is that not only was it the retinotopic order, where you have different cells responding to different parts of space, but also the hierarchical structure was supposed to mimic what happens in the ventral visual stream. And in fact, the early papers using these sorts of networks were very clear that they were trying to model this hierarchical, spatially organized structure in the brain. So now, of course, there's many differences. If you look at something like AlexNet, one of the immediate differences is that whereas in the brain, there's constant recurrent connections, and there's tons of feedback connections as well. In fact, in the brain, you find that there are more feedback connections from a higher order region to a lower order region than the other direction. In something like AlexNet, of course, you don't have those feedback connections and you don't have recurrent connections. But nonetheless, at a very simple level, it's trying to approximate something like what happens in the brain. Okay, now, so that's great. So these models were designed with the brain in mind to some extent, but does that mean they actually have anything to do with the brain? Well, one of the things that's come out over the last 5 years or so that has surprised the neuroscience world and angered some neuroscientists, you know those internet ads where it's like this one crazy trick, you know, solves your dermatological problem, doctors hate this person. There's been a little bit of that with respect to deep learning in neuroscience because what has happened is that it's been shown that deep neural networks that were not designed with the brain in mind necessarily, things like AlexNet, for example, or VGG, stuff like this, it turns out that these networks end up fitting the data that we see when we record from neurons in the brain better than any of the models that neuroscientists had developed to date. And so what I'm showing you here is a plot from a paper by Nicholas Krigas-Corday and Kali Grazavi from 2014. So this is 5 years ago. And this has been replicated by multiple groups now in multiple different centers around the world. What we're plotting is a plot that shows on the x-axis the categorization accuracy of a model on an image data set. I think it might have been ImageNet. And on the y-axis, what we have is a measurement of the similarity between the activity patterns in the model and the activity patterns that you record in the brain, either of a human or a monkey. And each of these little dots corresponds to a different model. The black dots correspond to various neuroscience models that had been put forward by neuroscientists to try to explain the ventral visual stream. The colored dots refer to the layers of, I think this might even just be AlexNet, the layers of a deep convolutional neural network. So L1 is the first layer, L7 is the final layer. And remember, inferotemporal cortex, just to go back a moment, is the top of our ventral visual hierarchy. And what the researchers found, first of all, is there's an interesting correlation between the accuracy of models on classification of these images and the match to the representations in monkey and human cortex. And of course, the other thing that stands out is just how much better a match the higher layers are of the deep net compared to the models that neuroscientists had put forward. And this has been replicated also in the auditory stream and in other parts of the visual stream. And it seems to be a solid result. So we're now in this funny situation where these deep neural networks that were developed to try to solve a computational task, but developed with the brain in mind, are now doing a better job of fitting the data that we neuroscientists have been recording for a while than many of the models we've developed. Yeah, go ahead.
Okay, I'm a little bit confused.
Sure.
I kind of get the idea that the architecture matches.
Yes.
And even when it doesn't match, you're trying to say something about the activity, but I'm not sure what the activity of a neural network, what does that mean? And for that matter, even the activity of neurons, is that like averaged over some time window?
Yes, right. Okay, so the activity of the neurons is averaged over a time window. That's right. So what the experimentalists will do is they will record activity from the neurons, and they will take the average activity during the presentation of an image to the animal or the person. The other thing that they do in order to make the comparisons possible, which I'll just quickly go into here, the way they make this comparison is they look at the activity of the neural network or the brain in response to each of the images, and they construct a correlation matrix. So basically, how much does the activity of image 1 correlate with the activity pattern for image 100, et cetera, et cetera. You do this for all of your images, and then you look specifically at, in fact, the difference in the correlation matrices between a model and the brain. And that distance metric, which they call they refer to here as the representational geometry, basically, tells you how similar the activities are. So what this really tells you is that if I show a neural network an image of a car and then an image of a bike, I'm going to get a certain distance in activity space for the neural network. And then I do the same for the monkey, and I get a certain distance in activity space for the neural network. The distances in activity space for all of the images in the neural network are a better match to the distances in activity space that we see in the monkey's brain or the human's brain. Yeah?
Can this be used to figure out questions like, is the neural network really understanding something? Like if the monkey doesn't know that a bike is related to a car or their vehicles, but a human does.
Right.
The model correlates more with the human in that term. And you say that the model is understanding something.
So understanding is a difficult word. I think that typically when we refer to understanding, what we mean is that there's a whole host of other additional implications regarding that concept. And so does the neural network understand it? Most certainly not, because the neural network's not going to make any extrapolations about the nature of bikes and cars, nor will the monkey. The people probably do, though. Now, it's important to remember that there are other parts of the brain where the activity patterns will be very different from what you see in a deep convolutional neural network. And those would be the parts of the brain that are really responsible for these sorts of common sense links. Yeah?
Sorry, me again.
Yeah.
All right, so the activity matches or the distances between them.
Yeah.
But that still kind of seems to not tell us anything about the learning algorithm.
So let's go right from that question. So this is all very cool, but one of the things that really drives neuroscientists nuts about it is precisely that they don't like the learning algorithm that was used to train these networks. And in particular, what they don't like is backpropagation. Now, so let's talk for a minute about backpropagation and why they don't like backpropagation. Okay, so the key feature of deep learning, like it's a funny thing, but often deep learning is not defined. Did anyone at the start of the summer school like try to define deep learning for you? Okay, what was the definition you received?
There was a non-linearity, linearity, and there was a loss.
Okay. Those are true, but okay, let's be clear. The key feature of deep learning is that you have a hierarchical neural network. It doesn't need to be nonlinear necessarily, for the record. Sometimes people refer to deep linear networks, but it has to be hierarchical. And the other thing that makes it deep, really, is that the learning algorithm, the optimization that you apply to it, is end-to-end. So what that means is that if you have some loss function, you can say that every change you make in the network is helping you to reduce that loss function. So even in the lower layers or the upper layers, wherever you are making changes that help you reduce that loss function. It's end-to-end. And this is what distinguishes deep learning from shallow learning techniques. If you think about nonlinear kernel methods, for example, you have a hierarchy. You have some features that you project, you project your data up into some feature space, and then you learn on that feature space. But what you don't do is you don't optimize your kernels or your features in the lower layers in order to minimize your loss function. All of the optimization of your loss function is done at the final step. So what distinguishes deep techniques from shallow techniques is that the optimization is end-to-end. Now, to do end-to-end optimization, you need to be able to do something called credit assignment. Credit assignment basically is a way of determining for each of the neurons and synapses in your network what was their credit, or blame if you will, for any contributions to the loss function. And this is difficult potentially because if you have even just a neural network with one hidden layer, the contribution, say, of this synapse here to your loss function depends on all of the downstream synapses. And so that's why you have to employ a technique that can take account of these relationships through your hierarchy. The most obvious way to do this, to do credit assignment that is fully hierarchical and end-to-end, is backpropagation. As Jürgen Schmidhuber is fond of pointing out, backpropagation is both a very old idea and a very obvious one. Because basically what you say is, okay, I need to know what the contribution of this synapse is to my loss function. So what am I going to do? I'm going to take the derivative of my loss function with respect to that neuron or that synapse. That is literally the most obvious way to calculate its contribution to the loss function. So here we've got a simple neural network sketched out with input X, hidden layer activity H, and output Y. And then we're receiving some target. So just to keep it simple, I'm going to talk about supervised learning throughout this talk. But I want to be clear that everything I talk about could equally be applied to unsupervised learning or reinforcement learning, because the credit assignment problem still holds in these cases, and the basic calculations you have to do still are the same ones. But mathematically, it's going to be easier to just talk about supervised learning. So, okay, I've got some target, and I want to make my output match my target. So I want to minimize the error, the difference between my output and my target. So here are the equations that govern my neural network. This is my linear operations. So I've got synaptic weights W0 for the hidden layer and synaptic weights W1 for the output layer. And my activities are just nonlinearities applied to those linear operations, all the stuff you've seen. My error is just the difference between my output and my target, because my loss function then I'm going to just define as the squared error, standard stuff. And now my update for my synaptic weights is just going to be gradient descent. So I take the partial derivative of my loss function with respect to these weights, W0. That's going to be the update for the weights. And then I use yield chain rule to work out all of the terms for that. Great, done. And as you know, this works surprisingly well. So let's just go through for a moment what you actually need to do this then. So here's the equation that I get. These are just each of the partial derivatives, basically, that I get from my chain rule. So I've got my error. And this error, notably, is the difference between my output and the target. And that output has to be just what the network does by itself when activity propagates forward through it. Okay? So I need the difference between my output generated by a forward pass and the target. I also need, because of the partial derivative of the downstream activity, the transpose of the downstream weight matrix. So remember, I'm updating W0 here. Just to go back one moment. This is the equation for updating W0, these synapses here. W1 is these synapses here. So what I need to do the gradient descent for W0 is I need the transpose of W1. You probably don't have to do this anymore, but when I was a student first learning this stuff, you had to like explicitly hand code this into your models, right? Now you just give it to PyTorch or TensorFlow and it's great. But if you were actually going the guts, you'd see that PyTorch and TensorFlow were using exactly this sort of information. Okay, and then the other thing we need is, in addition to the forward pass, we are basically passing this error backwards through the network. So the forward pass, we need not only the output, but we need the hidden unit inputs, the linear sums to the hidden unit inputs, and the derivative of my activation functions with respect to those. And at the same time, I need my error, and I need to send that back to my other neurons. So the problem is that basically every one of these items is biologically problematic in some way or another. The error term, the transpose of the downstream weights, and the separate forward and backward passes required to calculate all of these things are not things that we see in the brain. None of it really fits with what we know about synaptic changes in the brain. And so for that reason, neuroscientists from the 80s onwards were basically like, backpropagation has nothing to do with the brain. So all of your models that you train up with backpropagation have nothing to do with the brain. So please stop talking about it. And the fact that these models then fit the brain better than many of the models that we've developed based on our biological knowledge has caused us to kind of revisit the question, though. So in fact, what we've seen over the last few years is that although each of these terms looks problematic from a biological perspective right off the bat, there are actually ways to do end-to-end optimization to approximate the gradient of your loss function without utilizing these terms. And that means that even though the brain doesn't do backpropagation, and I want to emphasize that, I am not saying the brain does backpropagation, because it doesn't. It means that there are other ways to do credit assignment that are biologically realistic. So I'm going to show you those now. Okay, let's start with the error term. So one of the things that is difficult is precisely this error term. So although the brain sometimes calculates errors, and we see that, so it's not biologically implausible to assume that the brain has calculated an error. What's biologically implausible is the idea that errors are the things that are determining your synaptic updates. And the reason that seems implausible is precisely because we've seen in thousands and thousands of experiments biologically that you can get synaptic changes without an error occurring necessarily. Whereas if you actually look at the backpropagation algorithm for supervised learning anyway, there has to be an error. If there's no error, there's no weight update. Now, in fact, what we see biologically is that the key thing that determines whether or not neurons change their synapses is just the presence or absence and the timing of something called spikes. So for those of you who are unfamiliar with neurons, I had previously glossed over what happens in the axon, that output branch for neurons. What happens in the axon is something called an action potential or a spike, which is what's being illustrated here. So on the y-axis, we have the membrane voltage of the neuron, and the x-axis is just the extent of its axon. So this is its axon here. And when the neuron gets sufficiently activated, such that it hits some, what we call the spike threshold, the voltage will engage in this nonlinear depolarization, this rapid shoot up, and then a subsequent swing down. And that shoot up and swing down of the voltage will propagate along the axon. This is what we call a spike. So spikes will originate at the cell, what we call the axon hillock, and then they will propagate down the axon. And what researchers have shown is that the timing of spikes in the presynaptic and the postsynaptic neuron seem to be critical for determining synaptic plasticity. So this is a phenomenon known as spike timing dependent plasticity. It's been replicated again by countless experiments. It's very well established. This is an illustration of the basic phenomenon as it was first described by neuroscientists a couple of decades ago. So essentially, what you've got on the x-axis is the difference in timing between the spike of a presynaptic neuron and a postsynaptic neuron. So the neuron sending the spike is the presynaptic neuron, the neuron receiving the spike is the postsynaptic neuron, and it can spike itself, okay? So we're looking at what is the difference in timing between the spike that the presynaptic neuron generates and the spike that the postsynaptic neuron generates. 0 means they spike at exactly the same time. Positive means that the presynaptic neuron spikes before the postsynaptic neuron. And negative means that the postsynaptic neuron spikes before the presynaptic neuron. And so what neuroscientists would do is they'd run experiments where they would electrically control two neurons that were synaptically coupled, and they would force the neurons to spike at particular times relative to each other repeatedly. And then they would look at the change in the synaptic strength between those two neurons after they had forced them to spike repeatedly at this particular time window. And the results are plotted here. So here's what synaptic change you get depending upon what difference in timing you force on the neurons. Now this is biology, so it's very noisy. Each dot represents an individual experiment, but we can kind of fit it with a rough curve. And what you see is that you get potentiation of your synapse if the presynaptic neuron spikes just before the postsynaptic neuron, and you get some depression of your synapse, so a weakening of your synapse, if it spikes just after it. And so when this was first described, neuroscientists got really excited. They were like, okay, we've figured out the learning algorithm of the brain, great. And if any of you know the Blue Brain Project or the Human Brain Project, basically one of the premises of that project was we already kind of know the learning algorithm of the brain. We're just going to simulate 80 billion neurons, stick spike timing dependent plasticity in it, and go, intelligence will emerge. Most neuroscientists knew that was bullshit, but yeah, go ahead. Yeah. Okay, so again, I could teach a whole lecture on these questions, but short answer is you can measure the synaptic strength by causing the presynaptic neuron to spike and then measuring the voltage change that you see in the postsynaptic neuron. So what we're really measuring is how much does the voltage change in the postsynaptic neuron when the presynaptic neuron spikes. The question about other changes, yes, there are many other changes that can occur, but one of the things that has been established very well and fairly well in neuroscience at this point, and people still argue about it, but we're literally like decades in, so I think the discussion's starting to be more settled. When animals learn, you see synaptic changes. If you stop synaptic changes, they don't learn. If you force synaptic changes, they do learn. It looks like synaptic changes are critical to learning. There are probably also other changes that are happening, but roughly, yes, synaptic changes seem to be critical to learning and memory. Okay, so neuroscientists say, well, look, this is the learning algorithm. This is how neurons change their synapses. So they're not following an error gradient. They're just paying attention to temporal correlations in spikes. This is what we call Hebbian plasticity. It's named after a Canadian neuroscientist from Montreal, Donald Hebb, who first postulated that that's going to be the synaptic plasticity rule, is basically correlations in activity. But, of course, STDP by itself is not following a gradient of a loss function. And to get end-to-end optimization, you need to follow the gradient of a loss function, or at least estimate it. So Joshua and his postdoc, Benjamin Scalier, back a couple of years ago, proposed this algorithm called equilibrium propagation. The idea behind equilibrium propagation is as follows. So here we've got a neural network with some input and some output, x and y, so similar nomenclature to what I used before. And again, we're going to do this in the supervised framework, but remember, it could be applied to other frameworks. And so we're going to receive a target D. That's the one difference in variables here. D is our target. So we want to get y to match D when we give it a particular x. But this is going to be a fully connected neural network. Now, they're going to use symmetric weights here, and this is one piece of biological implausibility, but we'll come back to the question of symmetric weights in the next bit. But they're going to use symmetric weights here, so they've got a recurrent neural network with symmetric weights. U refers to all of the activity in the neural network. S refers to only the activity of the hidden units and the output units. And then sigma is our activation function. Now, what they do is they introduce this additional term beta, which refers to basically, so the targets here are, we're not going to take the difference between the targets and the output. Instead, the targets are just going to activate the output neurons a little bit. They're going to nudge them in a particular direction. And the extent to which they nudge the neurons is going to be determined by this variable beta. So when beta is equal to 0, our target neurons, D, are not having any impact on the network. When beta is greater than 0, they are partially clamping the output neurons. Now I say partially clamping because they are not, it's not that we are setting Y to D, it's just that we are allowing D to influence the activity of Y. So it's pushing the Y neurons in a particular direction. And what Benjamin proved in this paper here, was the proof in this paper? It might have been in another paper, but anyway, he proved it, and you can read about it either here or in the paper reference here, is, I think it was in here, that if you define the following weight update, you actually get something that is following the gradient. So it's not quite backprop, but it's an estimation of backprop. Yep. Yes. No, it means that if neuron A connects to neuron B with strength W, neuron B connects to neuron A with strength W. Yes, so you've got fully connected feedback and feed forward, and the feedback neuron, the feedback synapses are all equal to the feed forward neurons. Synapses rather, sorry. So what they do is they take basically just the correlation, they take the multiplication of the activity of the hidden units and output units during the phase when the target is influencing the output, and during the phase when the target is not influencing the output. Now, to be clear, they have to let the network settle. So what they do is they put the target on, or they don't have it, and then they let the network settle to a particular equilibrium distribution, and then they take these products. And if you take the difference of these products, I'm sorry, I'll get to your question in a moment. If you take the difference in these products between the weakly clamped phase and the non-clamped phase, in the limit where beta goes to 0, you actually fully recover backpropagation. Now, what's interesting about this is you're doing backpropagation without propagating an error. It's not that you have to calculate an error and then send it to all the neurons and say, okay, update your synapses if you have an error. Instead, you just take correlations in activity, And you're going to take correlations and the difference in correlations and activity between two different time points, and that's how you're going to follow your error gradient. Sorry, you had a question. Sure. So it basically just means that you are going to let y move closer to the activity pattern that d tells it to be. So you can think of it as literally just like if this is the y without d, here's d, y is going to do that. In Euclidean space, it's going to move towards d a little bit. That's right. You got it. Yeah. Yep.
So I still don't understand what is the phase state. Is D and Y time series data or
Is d and y?
time series data?
No. So here we're talking about, this is an algorithm for static supervised learning. So you can do things like classify images with this. So X could be an image and D could be the classification targets for that image, for example. So the phase is, you present the image, you let activity propagate through the network, but you don't let Y be influenced by D. That's your free phase. Then you do another phase where you present the image, you let activity propagate, but now you let D push Y towards a particular value. And you let the activity settle in the network towards an equilibrium distribution, and you take these products. Yep. Yes, basically, if I understood your question correctly. So what's happening essentially is every neuron's paying attention to its correlations with its neighbors during the phase when the target was influencing the network and during the phase when the target wasn't influencing the network. And the difference in those correlations between those two phases is your error gradient. This is reminiscent of if you've ever seen Boltzmann machines. This is basically the same thing as Boltzmann machines, okay? Sorry, you first and then you, yeah. I'm sorry, can you speak up? I can't hear you. I mean, that's what they assumed in this paper. I don't know whether the proof requires that. I suspect it wouldn't necessarily, but I'm not sure. Sorry, I'm sorry, I'm having trouble hearing you. Please speak up again. Oh, well, yes, because when you look at activity in the brain, your ability to control your arm depends upon how close in Euclidean space you get to the right activity patterns for your motor neurons, right? There isn't a fancy metric for your motor neurons, right? Like whether or not I successfully bend my elbow like this depends on the Euclidean difference between the activation command required to do that and what I actually do. The closer I get to it in Euclidean space, the closer I am to the right movement. So I suck at basketball because I am never anywhere close in Euclidean space to the commands that are required to shoot a ball into a basket. Yes, go ahead, and then I do have to move on, yeah. That's right.
Where these dynamics are given by discrete and time phases.
That's right.
But then the question is, how do you, like usually these dynamics depend on the initial state.
Well, this is why you have to let it settle to the equilibrium distribution.
Yeah, but even if you let it settle, depending on where you start, might have that one different position.
No, so the way that the networks are set up, you will eventually start sampling from an equilibrium distribution. They are Markov chains that will eventually settle on an equilibrium distribution. Now, the reason I went into this after telling you about STDP is because what's interesting is that another thing that Joshua's group showed was that if your weight changes follow this equation, so remember U is the activity of your hidden units and output, if your weight changes follow this equation where it's the temporal derivative of U for your postsynaptic neuron multiplied by the activity of your presynaptic neuron, then if you run one of the experiments that the neuroscientists do when they do STDP experiments, you get out the STDP curve. So what that means is that you can have a learning algorithm that's doing gradient descent, but if you do this very highly artificial experiment where you force activity to occur at particular times in pre- and postsynaptic neurons relative to each other, you end up recovering the STDP curve. Now, note that this is not the learning algorithm that Joshua and Benjamin proposed, but you can show that their learning algorithm adheres approximately to this equation. So that's one of the things that I think has confused neuroscientists a lot, is that they will see a particular pattern of synaptic changes, and they'll say, okay, this is the learning algorithm. But depending upon the specific activity patterns you do and the approximate equations, temporal dynamics that your synaptic changes follow, you will see certain things emerge that might not actually reflect your learning algorithm necessarily. And it also means that you can do gradient descent-based learning without propagating errors, using just correlations between neurons, and you will see things like what we see experimentally. Okay, so that is the error term. It's not as much of a problem as we thought. We can do gradient descent without explicitly passing around error terms in the brain. And it seems to, in fact, predict the experimental data we've seen on STDP. Okay, the next issue I want to talk about are the downstream weights. So remember, we had that term in our equation, W1 transpose. And this is biologically problematic, because what that says is, if I'm a neuron, in order to update my synaptic weights, I either have to know all of the downstream synaptic weights from me, which is totally not possible according to any laws of physiology we know, or I have to have feedback connections that are perfectly symmetric, which is what I just mentioned with respect to equilibrium propagation. So equilibrium propagation doesn't solve that problem. It still assumes symmetric feedback weights. So one way you can think about this is basically here. This is a sketch of a neural network. So we've got input, hidden unit, output. We're going to send our error back through some synapses. For every synapse in the feed-forward pathway, colored here, we need to have a corresponding synapse in the feedback pathway with the exact same value. In other words, we need a feedback pathway that is equal to the transpose of W1. If you have that, you can do backpropagation in a reasonably biological way. The problem is that the assumption that your feedback connections match perfectly your feed-forward connections is not something the brain can guarantee. So this was another reason that biologists and neuroscientists said, look, backprop has nothing to do with the brain. Forget about it. Now, my friend Tim Lillicrap and we had long ago drank Jeff Hinton's Kool-Aid and had convinced ourselves that there was something about this backprop that might actually be relevant to the brain. And back in the, around 2012 or 2013, Tim was trying to think about ways that rather than forcing the network to have these symmetric weights, you could learn it. So you could have a learning algorithm on your feedback connections that would encourage it to develop symmetry and therefore let it do backpropagation without having a hard constraint of symmetry. Now, in training his network, he wanted to have a control case. So he had cooked up this learning algorithm for the feedback weights, and he thought to himself, okay, what should my control case be? Well, my control case should be that I have some random feedback weights, B, that I set, and then I fix them. So I don't let the network update its feedback weights. I'm going to propagate my error back through these random feedback weights and use that to update the synapses in the hidden layer. And if I do that, learning should be garbage, and then I can show that my learning algorithm is going to be great, and it's going to do like backprop. What was really weird, though, was that his control network learned really well. So unfortunately for his ultimate goal, but fortunately for kind of scientific exploration, he found that when he trained with this random feedback network, his first test was, of course, MNIST, he actually got exactly the same level of error that he got with backpropagation. So he called his algorithm feedback alignment for a reason that I will explain in a moment. And as you can see, when you train on MNIST here, you have basically the exact same learning curve for backpropagation as you do for feedback alignment with a fixed random weight matrix. Now, here in gray is a shallow network where you don't update the hidden weights, okay? And shallow networks don't do as well on things like MNIST or ImageNet or whatever. So that's unsurprising, but this green curve was really surprising. Now it turns out that what's happening here is, so here what Tim plotted was, he plotted the angle between what his feedback alignment algorithm was proposing for the synaptic weights and what backpropagation was proposing for the synaptic weights. So basically, think about your weight updates as a big vector, okay? So you take all of your weight updates, you make a big vector, and you've got two different algorithms, feedback alignment and backpropagation, and you ask, what's the angle between those two vectors? Now, if you take two random vectors in a really high dimensional space, they're going to be orthogonal to each other, 90 degrees. And at the start of learning, that's exactly what Tim found. They were 90 degrees to each other. But over the course of learning, the angle between them shrunk down to below 45 degrees. And one of the things that's maybe a little bit counterintuitive, but is important to remember, is that in a high dimensional space, 45 degrees is basically walking in the same direction. So this meant that the algorithm was actually kind of doing backpropagation after the first few epochs. Now the reason was, it turned out that the feed-forward matrix, W1, was aligning itself to that random feedback matrix B. And that's why he called it feedback alignment. And this has been replicated again and again and again and again. It turns out that in certain circumstances with certain tasks, and I'll show you a case where it doesn't hold, you can basically always see that the feed-forward weights will in fact align themselves with your feedback weights, whatever they are. And that means that you can actually follow the gradient of an algorithm without having that explicit guarantee of symmetry. Now, Tim and I have been exploring this more. One of the things that we find, unfortunately, is that depending upon the network architecture and depending upon the data set that you're training on, it seems to be more difficult to see this feedback alignment effect occur. So with MNIST, it's super easy. It always works. If you train on ImageNet, though, and in particular if you train with convolutional structures, it can be more difficult. So here was our paper from NeurIPS last year where we show that basically when you look at training in a deep convnet for feedback alignment, and actually even not a convnet, if you just take a fully connected network, feedback alignment versus backpropagation, there's no comparison. Backpropagation really beats it on ImageNet. And especially, it doesn't even come close to working with convolutional architectures. The reason is probably that, this is my intuition, but no one's proved this for the record. Depending upon the network structure that you set up and the data set that you have, you are limiting the space of possible feed-forward architectures that would, feed-forward weight matrices that would work for solving your task. And the issue is that the feed-forward matrices need to be able to align with the backward weights and solve your task. If it just so happens that the alignment doesn't correspond to one of the potential solutions for your task, or if you really constrain your feed-forward weights, e.g. with convolutions, then this feedback alignment effect doesn't seem to work well. Yeah? Yeah, if you massively over-parameterize, it works well. Yeah, because then you can let it align well. But another possibility is Tim's original idea. So I think the important thing for feedback alignment is to realize that you don't need a perfect guarantee of symmetry. The network can learn to align itself, but probably what you want to do is not keep your feedback weights perfectly fixed, but do some learning there as well, as per his original idea. And then the feedback weights and the feed-forward weights can work together to try to settle upon a solution. So one idea that Tim and his friend Doug Lillicrap came up with recently was to go back to a very old algorithm known as the Colin Pollock algorithm from the 90s. Basically, Colin and Pollock noted, okay, let's say you have two weight matrices, totally randomly selected initially, but what you do is every update, you update them with the same update, A, and then you apply a bit of weight decay. So my synaptic updates are always going to be the same term and some weight decay. If you do that, you're guaranteed that your two matrices are going to come into alignment. And this is something that you could theoretically have happening in biology, because your presynaptic neuron and your postsynaptic neuron have access to the same information. So any updates that you're doing in your postsynaptic neuron, your presynaptic neuron will probably know about. And it means that you are basically able to say, okay, I want my synapses to have the same update plus a bit of weight decay, which is super simple biologically. So using the Colin Pollock algorithm is a perfectly reasonable proposal for the brain, and it works nicely. Here's training on ImageNet, and you can see backpropagation there in black and the Colin Pollock algorithm in red. So we can get back to backpropagation level performance on tough tasks in convolutional neural networks if we give the feedback alignment effect a little bit of help using the Colin Pollock algorithm. So again, it means that this need for perfect weight symmetry to be guaranteed is not the case. So we have, yeah, I've said that. 2 out of 3 items down. Turns out you don't need the symmetric weights. Okay. Now the last thing. So as I mentioned, there's this problem of the forward pass and the backward pass. Like the way that backpropagation was originally conceived in terms of how it could potentially operate in the brain is that you have a forward pass of information through the network, the errors get calculated, and then the errors get backpropagated through the network, hence the name. The difficulty is that this is not what we see in the brain. Despite those hoary old adages about like 10% of your brain or something like that being active at a time, the reality is your entire brain is constantly buzzing. And you don't, although you see in anesthetized animals some interesting evidence for like a sort of feed forward phase and then a feedback phase, in an awake animal, you don't see good evidence of this. Basically, when an image is presented to an animal, it will alter the activity in the brain, but it's not that the activity propagates through, say, the ventral visual stream and then gets propagated back downward. So theoretically, at least, we have no reason to believe that the brain engages in a forward pass followed by a backward pass. But of course, real neurons, especially those in the neocortex, well, pretty much everywhere, but especially those in the neocortex, which is the brain region that we were talking about with the ventral visual stream. It is the seat of higher order cognition, and it is what differentiates mammals from other species. The neurons in the neocortex are far more complicated than an actual just linear sum followed by an activation function, despite what I told you earlier. So that's how they behave. They behave linear followed by nonlinear activation for a lot of what they do. But occasionally you see them engage in some really interesting nonlinear computations. And in particular, the place that they seem to engage in a lot of their nonlinear computations is in their dendrites. And there are different nonlinear computations in different types of dendrites. So here on the left, we have an illustration of a pyramidal neuron. It's called a pyramidal neuron because its cell body is kind of shaped like a pyramid. And pyramidal neurons they actually have 3 different sets, but I'm not going to go into that. They have 2 sets you need to know about of dendrites, called the basal dendrites and the apical dendrites. The basal dendrites come out from around that pyramidal cell body. And the apical dendrites come out from this long branch that the neurons send up towards the top of your brain. So this is where the surface of the brain sits. The cell bodies will be embedded deeper in your neocortex, and they will send these apical branches up to the top of the brain. It's kind of shaped like a tree. So the basal dendrites are like the roots of a tree, and the apical dendrites are like the branches of the tree. And the cell body sits at the base of the tree with the roots. Now, here are some actual images of pyramidal neurons that my student Matt Tran took. So these are neurons from a mouse's brain, a mouse's neocortex, that have been filled with a fluorescent dye. There are 4 pyramidal neurons that you can see easily. Well, there's actually 6, but 2 of them are very faint. So there's these 4 neurons here. This one's the clearest one. So here's its cell body. This is its axon. So you can see that axon heading out there to communicate to all the other neurons. Here are its basal dendrites, and here are its apical dendrites up at the surface of the mouse's brain. And one of the things that's really interesting is that anatomists have shown that different inputs are received at these different dendrites. So when you look at the inputs that are received at the basal dendrites, they tend to be feed-forward. They tend to carry the incoming sensory information from the retina or the touch receptors or whatever. In contrast, the apical dendrites seem to be the major site of feedback from higher order brain regions. So this means you've got this segregation of your feed-forward and feedback pathways into different dendrites in the brain. And I'm going to tie this into something meaningful in a moment. Just keep that in mind for a second. Now, the apical dendrites have this very interesting nonlinearity to them. So in that branch, the big, trunk as it were, they have these highly nonlinear calcium ion channels that allow the apical dendrite to engage in nonlinear computations. And in particular, what a researcher named Matthew Larkham showed back in the late 90s and early 2000s through a series of very difficult to run experiments. So what he would do is he'd attach an electrode, multiple electrodes to an individual pyramidal neuron, and he'd look at how voltage changes propagate throughout the neuron. So here we've got different colors of electrode, color-coded, okay? And here we see what he saw on his electrodes when he injected current into his red electrode. So what he found is that if he injected just a little bit of current into the red electrode, he would see a response in the red electrode, obviously, but only minimal responses further down the tree. Generally, the apical dendrites what he found was they'd engage in some linear summation, but whatever that linear sum was, it wouldn't actually get communicated to the soma, which was really weird. Why would you take the linear sum and then do nothing with it? The answer turned out to be that it does do something with it, but only if there's sufficient activation received at the apical dendrites. So if you stimulate the apical dendrites enough, you get what's called a nonlinear plateau potential. This is almost like a spike in the dendrite itself. It's this big upswing in the voltage that occurs, and when it occurs, it propagates fully down the dendrite. So it's almost like there's another neuron at the top here of the cell. And when it does that, when it engages in a plateau potential, it will not only drive the cell to spike, which you see in black here, those are 2 spikes, it will tend to drive the cell to spike multiple times in high frequency, which is something we call a burst in neuroscience. So the apical dendrites, given sufficient depolarization, will drive what we call high frequency spike bursts. Now what's interesting about this then is because the top-down signals come to the apical dendrites and the bottom-up signals come to the basal dendrites, and the basal dendrites tend to trigger single spikes, you've got this funny dichotomy in the activity of the neuron. If the neuron emits a single spike, it was probably driven by the basal dendrites alone. But if the neuron emits a burst of spikes, it was probably driven by at least both the basal and apical, definitely the apical. So that means that depending upon whether you get a single spike or a burst of spikes, you know whether you're receiving top-down feedback or bottom-up signals. And this means that you can simultaneously broadcast feed-forward and feedback signals in your pyramidal neurons without having separate forward and backwards passes. Because basically, your bottom-up signals can come through and they can be propagated with single spikes, and your top-down signals can be propagated with bursts. So this is an idea that Richard Node, my collaborator at University of Ottawa, and I have been looking at. So here we've got a simulation of pyramidal neurons. This is actually a biophysical simulation. And what we're measuring here are different things we call the event rate, the burst rate, and the burst probability. So the event rate is the rate at which we generate either a spike or a burst. The burst so we just call either a spike or a burst an event, okay? And then we can ask the question, what is the probability that an event in a neuron is a burst? That's the burst probability here. So you can measure then the event rate separately, the burst rate separately, and the burst probability separately. And what we can show in our simulations is that, according to the rules of the biophysics of pyramidal neurons, a top-down signal into the apical dendrites should be reflected in the burst probability of that neuron. So in the dotted line, you see the input to the apical dendrites scaled, and the solid line is the burst probability of these neurons, and you can see it's tracking the top-down input to the apical dendrites. Meanwhile, the event rate will tend to track the input to the basal dendrites. So here we see some input to the basal dendrites, and we see the event rate, and it's following that. The burst rate is then going to be a combination of those two. But what we can show is that if you communicate the burst rate down through the network to another set of neurons into their apical dendrites, and the event rate up to another set of neurons, you can basically send your top-down signal back down through the network. So here is in the yellow dotted line is the burst rate of the pink population of neurons, and here is the burst probability of the green population of neurons. And you can see its burst probability is tracking the burst rate of the upper neurons. And this is all happening in continuous time. So the neurons can communicate feedback to each other and propagate feed-forward signals up to the other one, all simultaneously using this multiplexed system. And what we can do then as well is we can use the difference in burst probabilities over time as our error signal. So this is a little bit akin to what they did with equilibrium propagation. The idea is that we're going to look at the burst probabilities at a particular time, and in fact we just take a time step. So the burst probabilities at t plus 1 minus the burst probabilities at time t. That difference we can demonstrate actually carries a gradient signal if you've propagated a target down through the network. And then we just multiply that with these event rate terms, and we can show that this synaptic plasticity rule, which is all perfectly biologically plausible, will actually follow a gradient. Now here's what it actually looks like if you kind of did it experimentally. So here we're plotting for, these are the postsynaptic neurons, these are the presynaptic neurons, the burst rate and the event rate, and the somatic weights, so the weight, this synapse here. And what you can see is that essentially the synapses change according to what the burst rate does. So when the burst rate increases, the synapse increase. The synaptic weights increase. And when the burst rate decreases, the synaptic weights decrease. And this can actually be demonstrated experimentally as well. So people have shown that STDP is particularly sensitive to bursts. And you will get sign switches depending upon the particular, whether or not you have a burst in your postsynaptic neuron. Now, note though that it's not tracking what's going on in the lower neurons. So the lower neurons' burst rate or burst probability is not doing anything interesting. So that means that you can do a top-down control on the synaptic plasticity in your network. I probably don't have the time to go into this stuff, so I'm just going to skip that. But this is basically just a demonstration that it replicates some experimental data. Anyway, long story short, we can show that we can estimate the gradient of a supervised loss function with this. So if we take MNIST or CIFAR 10, what we do here, now we haven't figured out how to do this in a biologically realistic way with convolutions. Because to be clear, one thing I didn't say previously, convolutions themselves are biologically unrealistic because of weight sharing. In a convolutional neural network, you're assuming that every neuron in space has the exact same synaptic weights. That doesn't hold in the brain. So that's a caveat that is still something to contend with, and I'm happy to discuss in person with you ways that we could potentially contend with this. So what we do in our network here is we have a series of convolutional layers, and then we have a series of our pyramidal neuron layers where we're simulating these pyramidal neurons with these bursts and spike signals. And what we do is we take this difference in burst probability so if at time t star we deliver a target to the output layer, so it's like equilibrium propagation. We nudge the output layer towards the correct direction in Euclidean space, and we take the burst probabilities throughout the network at the time just after the nudge was arrived and just before it. And what we do then is we take those burst probability differences and we feed that into the convolutional layer as if it was the gradient from the layer above. So we basically say to the convolution layer, okay, here's our calculated partial derivative, go with it. And what we show is that we can achieve the same kind of learning that you achieve with backpropagation, shown in red, with our system that's doing this with these bursts and spikes in the upper layers. And critically, it does this without a separate feed-forward and feedback phase. So using the principles of how the neurons are built in the neocortex, we can eliminate the need for separate feed-forward and feedback phases in our algorithms. So 3 out of 3 down. Now, the next step ultimately is to test these ideas in the brain. There are many other models that have been proposed for doing biologically realistic gradient descent that I haven't touched on here. We're in an exciting time. We now have realized that the complaints about end-to-end optimization are probably not valid, and that it's entirely possible that the brain does something like end-to-end optimization, i.e. deep learning. Furthermore, we have evidence that deep neural networks match the representations in brains really well. So it's possible, but I emphasize that word possible, it's possible that something in our brains is doing end-to-end optimization. And given the multitude of models that we have now for potentially how to do this in the brain, it's time to start testing this. And hopefully 20 years from now, we'll have an answer as to whether or not one of the models is correct, or maybe none of them are, and it was all just a laugh all along. Now, the one thing I'm going to end on here is just to say a major unresolved issue that I haven't touched on is backpropagation through time. So when you train a recurrent neural network, you not only do credit assignment through your hierarchy, you do credit assignment through time. So you say, okay, the neuron at time t1 was responsible for this amount of loss at time t50, or whatever. The difficulty is that all of the solutions I just discussed with you don't apply to backpropagation through time. because they would only apply if we could somehow send these sorts of things that I was discussing backwards through time. But we don't know of any way that, say, the brain could send bursts backwards through time. Doesn't seem to be a thing. So this is still something to be resolved. You know, and I think that it's non-trivial, but it's an important topic, and I hope that someone takes it up. Maybe someone in my lab will someday, maybe someone else will. I think figuring out how to do credit assignment through time in a biologically realistic manner is one of the most important issues to address in this area right now. Anyway, so to conclude, there's ultimately a good reason that deep learning has taken over AI, much to the chagrin of people like Gary Marcus, and that is that it works, okay? No one would be excited about deep learning if it didn't work. And the reason it works is, you know, people discuss it. It's a whole area of research. But I think I can say the following with some degree of confidence. It looks like end-to-end optimization is really important for training in high-dimensional spaces. Because think of it like the analogy I always give, at the risk of going a minute or two over time, the analogy I always give is, think about the game Hot and Cold. Do you know the game Hot and Cold? So like someone has placed something somewhere and you're searching for it and they shout hot if you're getting closer and cold if you're going farther away. Okay, so a lot of the algorithms that people have proposed for the brain or various other things are basically versions of hot and cold. Hot and cold works really well when you're in 2D space or 3D space or 10D space. Hot and cold is a bitch in a million dimensions. If you're going to be operating in a million dimensions, you want someone to explicitly be like, go that way. And that's what end-to-end optimization does for you. It says, go that way. That's the direction that's going to make things better for you. So does that apply to the brain? I don't know, but I suspect so. And I suspect that, you know, the reason that we all do end-to-end optimization in neural networks is because it's how you do learning in high D. And our brains are super high D. We have 80 billion neurons in the human brain. The idea that you could train that system without some kind of end-to-end guarantee seems unrealistic to me. But for the record, many neuroscientists would disagree with me. Nonetheless, even though our brains don't do backprop specifically, that doesn't mean that they don't do some kind of approximation to it. And as I've shown you today, we've had a series of models over the last 5 years that have demonstrated that this is possible. And so I'm going to leave you with the last following kind of cool, scary thought. If our brains are approximating some form of gradient descent, and we neuroscientists can figure out what those gradient signals are, then theoretically in the future you could have neural prostheses that take the gradient signals from an artificial deep neural network and send those gradient signals into the brain, and then you could do end-to-end optimization through your AI into your brain. And then you'd have a seamless link between you and your AI, which would be either cool or terrible, depending upon what you use it for. So that's that. And I'm over time, but I'm happy to have questions, and I'm going to be around for the next couple of days as well. So if you want to discuss, please come up and chat. That's it. Thanks very much. Okay. You were quick on the go, so go for it.
Do you have a theory for what role the CIFAR neuron plays in learning?
Yeah, good question. So there's another piece of biology I glossed over. One of the things you might not know if you're not a neuroscientist is something called Dale's law. Dale's law states that synapses in the brain are only either positive or negative. You cannot have a synapse that switches from positive to negative or negative to positive. And obviously the weights in synaptic networks do that all the time. So first of all, one of the things for learning that we have to figure out is how do you do learning with Dale's law in place? Because Dale's law turns out to be complicated for doing learning. The second thing though, which really more directly gets at what I think you're asking, is potentially the role of inhibition in gating learning. And so one of the interesting things that's been emerging experimentally is that there are specialized inhibitory neurons that target the apical dendrites of pyramidal neurons and which seem to control learning. If we turn these neurons on or off, we can gate learning in animals' brains, basically. Sure, you go ahead. Sorry, speak up, please. Yeah, so the question was, what about spontaneous activity? Is that helping us to learn? And the answer is, spontaneous activity could very well be helping us to learn. It in fact fits into the equilibrium propagation framework. Because part of what Joshua was thinking in his models and what people thought with the Boltzmann machine models is the idea is that the free phase is like spontaneous activity. It's like when you're dreaming and the environment's not impinging on you, telling you what things you have to learn. If you take the difference between your spontaneous activity and the activity that you get when the environment is forcing particular ways of seeing the world on you, that can actually be a gradient signal. Okay, we're done? Sorry, we're cut off. Yeah.